Hello, and welcome back to the Biology 219 video playlist. This week, we'll be talking about how we know where genes are in the genome. This is important. If we know where they are, we can practice reverse genetics. In other words, we can break a gene on purpose if we know where it is. And then see what happens to the cell's function. We can also see what happens when changes happen naturally in a population. There are many different alleles of each gene, and not all of them are bad. If we could sequence each allele, we could use those sequences to diagnose and possibly treat diseases. Knowing where a gene is depends on a really specific definition of a gene. We will define a gene as all the information necessary to make a single product. Humans are diploid, so there are two copies of each gene in the cell. In a homozygous individual, those two copies are the same allele. Alleles are variations of a gene. In heterozygous individuals, there are two different alleles. In a population of organisms, there can be many different alleles because there are many ways to vary the sequence of a gene. The idea of alleles goes back to early genetics and Gregor Mendel. Remember that Mendel was systematically breeding peas. He developed homozygous lines of pea plants and then crossed them together. He didn't know about genes or DNA, so he developed his principles by looking at the phenotypes from the offspring. For example, when he crossed yellow peas with green peas, all of the offspring were yellow. If he took those yellow offspring and crossed them together, 75% of the offspring were yellow and 25% of the offspring were green. From this, he derived three principles: the principle of dominance, the principle of segregation, and the principle of independent assortment. The principle of dominance simply states that when two alleles of the same gene are present, one can mask the presence of the other. In other words, one of the alleles can dominate over the other. A simple example of this is eye color. The default eye color is blue. But if a protein is made, like melanin, that has another color, like brown, the new color will be seen instead. The allele that codes for blue eyes is still there in the genome, but only the brown-eyed phenotype is seen. The principle of segregation says that alleles of the same gene are segregated during the formation of gametes. To understand this, we have to review meiosis. Let's imagine a person who is heterozygous at a locus A. Remember that meiosis starts in S phase, when each of the chromosomes is replicated. The chromatids will condense in prophase and align on the metaphase plate in metaphase 1. These will separate to the poles in anaphase 1, and will be divided into daughter cells in telophase 1. At this point, the different alleles have already been segregated into different cells. These cells will go through meiosis II, and each daughter cell will be haploid with only one copy of this gene. You could draw this in a Punnett square to predict the possible phenotypes when these gametes come together with other gametes during fertilization. The principle of independent assortment states that alleles of two different genes are segregated independently during the formation of gametes. Let's imagine our same person from the previous example, but now we'll watch what happens to our original gene A and a new gene B, which is on a different chromosome. The interesting part happens right away in metaphase 1. Each chromosome will align with its sister chromatid, but there are two possible outcomes. It could be that both dominant alleles, A and B are on the same side, and both recessive alleles are on the opposite side. Or it could happen that dominant A is paired with recessive B, while dominant B is paired with recessive A. These outcomes are equally likely, and which one will occur is completely random. You can see that as these continue through metaphase to anaphase and telophase 1, there are 4 possible outcomes by which these gametes could be combined, and A and B segregate independently. As the cell proceeds through meiosis 1 and then through prophase 2, metaphase 2, anaphase 2, and telophase 2, you can see that there are 4 possible combinations of alleles that can occur. You could see how these might combine with other gametes in a Punnett square, like this. Here again, looking at peas, there are 4 different alleles. On one gene, there's a dominant allele that leads to yellow peas, and a recessive allele that leads to green. At a different gene, there's a dominant allele that leads to round peas, and a recessive allele that leads to wrinkled peas. If 2 heterozygous individuals were mated together, you would receive a 9:3:3:1 ratio of phenotypes. Finally, independent assortment is only possible if alleles are on different chromosomes. Let's see what happens if they are on the same chromosome. In this example, dominant A and B are on one copy of the chromosome, while recessive A and B are on the other copy. When these replicate and sister chromatids align during metaphase, there are only 2 possible outcomes after division. Either the daughter cells will have 2 copies of the dominant allele, or they will have 2 copies of the recessive allele. There are only 2 possible outcomes because the alleles themselves are linked on the same physical copy of DNA. The homozygous alleles in this example will always travel together into daughter cells, and so will the recessive alleles. We will see in class that the concept of linked alleles allows us to figure out where genes are on a chromosome relative to each other. This is how the earliest genome maps were built. More recently, Mendel's principles have been used to think about evolution, medical diagnosis and treatment, and agricultural improvement.